According to the Department of Agriculture, Trade, and Consumer Protection, the dairy industry provides about $52.8 billion to Wisconsin's economy each year. That means that dairy cattle are producing just under half of Wisconsin agriculture's $116.3 billion economic contribution. Dairy farmers rely on each drop of milk to make not only their livelihood, but to ensure that they're meeting their customer demands. We know that mastitis is one of the most frequently diagnosed diseases that dairy cows experience. This infectious disease has a huge impact on dairy farms, economic impact on dairy farms, due to the rising costs in treatments, as well as the loss of dairy revenue as cows produce less milk during each case. Over the years, farmers and researchers have worked hard to develop not only prevention strategies, but treatment strategies. One of the most common treatment strategies today is the use of antimicrobials. However, oftentimes these treatments are not based on the etiology or the species of mastitis-causing bacteria. Farmers are facing many challenges over increased societal concerns over the use of antibiotics, especially with the growing crisis of antibiotic resistance. We know that different species of mastitis-causing bacteria could more easily be prevented rather than treated. Therefore, the objective of this study was to determine the frequency of different mastitis-causing bacteria on local Wisconsin dairy herds with low ball tank somatic cell count. We enrolled about 8 farms total at this moment. We look to get more. Farms that have less than or equal to 150,000 somatic cell counts in their bulk tank. Of those farms, we used their farm reports to locate cows that had greater than 200,000 somatic cell count on their last DHI test. Those cows were then sampled. We took 2 samples from each quarter. An aseptic sample was then used to isolate and culture the mastitis-causing bacteria, and the non-aseptic sample was sent to a lab in order to verify that each cow, in fact, had greater than 200,000 somatic cell count. The frequency of isolated bacteria was then calculated as a percentage, and our preliminary data showed that minor mastitis-causing pathogens may be more prevalent than what was previously shown in research, highlighting the importance of speciating mastitis-causing bacteria to enable more specific mastitis prevention strategies. One big idea of our research is how to better educate farmers on the use of different prevention strategies based on specific mastitis-causing bacteria versus continual use of antibiotics. Thank you.
All right, thank you very much, Rainna, sorry.